Good evening. What about only Padmavati and Sujata? What about others? Yes.
Sir, they will join, sir.
Yes, my voice is audible.
Yes, sir.
What about others, Amma? Only 2 members.
They will join, sir.
Okay. So what about before 2 days before we discussed the what are the chapters? Yes, any idea? Today we will discuss development of endosperms, right? Yes. Yes, tell me. Please respond. Endosperm of development of endosperm. Last week we completed, I think, fertilization, right? Right? What is the endosperm? Yes. My voice is audible?
Yes, sir.
What is the last class?
Sir, I was absent, sir, last class.
So what about other? What is endosperms? Yes. The endosperm is the nutritive tissue, right? Yes or no? Endosperm is the nutritive tissue for the developing embryo. Phanerograms means? Yes.
Flowering plants.
No. Wrong answer. Phanerograms. 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 These are the, in Telugu, what we call Vibruta Bijalu. There is no flowering. What is the angiosperms?
Flowering, sir.
Flowering plants. So here the endosperm is the role of and function of endosperm. It is the nutritive tissue. So nutritive tissue, what is the role of nutritive tissue? It is provide the nutrients for the developing embryo, right? Yes, clear?
Yes, sir.
Do you understand? What is the nutritive tissue? It is the nutritive tissue for the development of, yes, development of embryo tissue, right?
Yes, sir.
So here it is phanerograms. What is the role of endosperm in the gymnosperm? Here the gymnosperms, the endospore is the applied nature. What is the meaning of applied? Applied. Applied.
N, sir.
1N. 1N means one set of chromosomes. And it is formed in the before fertilization. Okay. So in phanerograms, it is the nutritive tissue for the developing embryo. It is gymnosperm. The endosperm is the haploid and is formed before the fertilization. So in angiosperms, it is a triploid. Triploid means 3n number, right? 3n. What is the meaning of 3n?
Triploid, sir.
Triploid. It is 3 sets of chromosomes. So here, the endosperms of angiosperms, the primary endosperm nucleus after fertilization. The angiosperm endosperm is the triploid 3n number nature, and it is developed from the primary endosperm nucleus after the fertilization. So in gymnosperms, the endosperms formed in the before the fertilization. Here, after the fertilization, what are developed? It is developed from the primary endosperm nucleus, right? And in most cases, the endosperm is triploid. Since the number of polar nuclei varies from different types of embryo sac, cytological nature of endosperms also shows some variations. Okay.
Yes, sir.
So, just wait. For example, Oniothera type of embryo sac, there are only one polar nucleus. The endosperm is diploid nature, and the Pepermoia type of embryo sac is 8 polar nuclei. 8 polar nuclei. Hence, the endosperm is in 9n. That means 9 set of the chromosomes. So endosperm is the characteristic of all angiosperms, with the exemption of Archidaceae, Trapezae, and Podostemonaceae. The endosperm is the characteristic of all angiosperms, with the exception of what are the 3 families? Archidaceae, Trapezae, and Podostemonaceae, right? In plants like beans, peas, the entire endosperm is consumed for the nutrition of the developing embryo. The entire endosperm is consumed for the nutrition for the developing embryo. The mature seeds yes. The entire endosperms is consumed for the nutrition of developing embryo. Thus, mature seeds are without endosperm, and such seeds are known as non-endospermic, and they store food in the cotyledons. You know cotyledons?
Yes, sir. Two
So,
cotyledons are found.
yes. That is 2 cotyledons, dicotyledons, 1 cotyledons, and monocotyledons. On other hand, the even mature seeds of the cereals and coconut, custard, it is retained endosperm. Such seeds are called endospermic. The endosperm present in the seeds is used in the establishment of the young seedlings. When the endosperm, it is useful for the young seedlings. When the seed started is the germination process, that endosperm, it is the endosperm, it is a nutrient value. The nutrients, it is transferred to the entire this germinating plant. Okay.
Yes,
So
sir.
here my slide is visible.
Yes, sir.
Yes, sir.
So first of all, before going to endosperms, first of all, already we discussed in the two male gametes. Two male gametes, it is polar or secondary nuclei. It is egg cell, the endosperm, triple fusion, the zygote. These are the process of double fertilization. Some of the plants, the before fertilization, like gymnosperms is the before fertilization, in the angiosperms after fertilization. Okay, so yes, the most important plant product on the earth for the human beings, 2/3 of all human calories come from the endosperm, like cereals, wheat, rice, maize, barley, and millets, and some of the oils, coconut, palm oil, palm and castor oil. Next, coming to types of endosperms. Types of endosperms. On the basis of their endosperms development, 3 types of endosperms have been recognized. The first one is the nuclear endosperm. Second one is the cellular endosperm. The third one is the fallopian endosperm. How many types of endosperms?
3 types, sir.
3 types.
What is the first type? What is the first type?
Nuclear endosperm,
Yes.
cellular endosperm,
Yes.
fallopian endosperm.
Fallopian endosperm.
Endosperm.
These 3 types, nuclear endosperm is the most common. The nuclear endosperm is the most common and found in about 56% of families of the angiosperms. How much of percentage?
56.
56% of the which one? Which plant group?
Nuclear endosperm.
Nuclear endosperm.
Which
Angiosperm, sir.
Angiosperms. 56% of the family of angiosperms. It is the most common. Okay. So next one, second one is cellular endosperms. It is occurs in the only 25% of the families, and helobial endosperms in 19% of the families. The first one is the nuclear endosperm. It is, what is the, how much percentage? 56. The cellular,
25%.
how much?
25%.
25%.
And helobial,
19%.
19%. Okay. These and the further the coming to cellular endosperms is mostly confined to the dicotyledon families. It is mostly so in the nuclear endosperm, it is 56% of family. The
Dicotyledons.
here the 25% of the cellular endosperm, it is in the dicotyledon families. In monocots occurs with only RAC and Lemanaceae family. These cellular endosperms present in the monocots only 2 families, RAC and Lemnaceae. And helobial endosperms is only monocotyledon families. Only monocotyledon families. Like grasses. All the grasses are monocotyledons.
Okay.
Right?
Okay.
So clear? Once again I repeat.
Okay.
The types of endosperms is the 3 types of endosperms are there. In their basis on the development, they are recognized in 3 types. One is the nuclear endosperm, cellular endosperm, and helobial endosperm. These nuclear endosperms is most common and found in the 56% of the families of the angiosperms.
Angiosperm.
And the cellular endosperms is occurs in the 25 families
25%.
in the dicotyledons family.
Dicotyledons.
In monocots, only 2 families.
2 families.
Araceae and
Arachis and
Lemnaceae. The
Lemini.
helobial endosperms present are found in the only
Grasses.
monocotyledon families. Clear?
Yes, sir.
Next, we go for nuclear endosperm. Nuclear endosperms. Already we discussed most common found in the about 56% of the families. So here, no cell wall formation during the initial few divisions of the primary endosperms nucleus, and nuclei are remain free in the cytoplasm of the embryo sac. So here, what happened is there is no cell formation. Generally, the formation of endosperms, the initial few divisions, like whatever divisions, mitotic or meiosis, whatever divisions occurs, it is initial few divisions of the primary endosperms nucleus. First of all, what we formed already in the discussion, in the angiosperm is plant in the triploid and develop from a primary endosperm nucleus. The primary endosperm is the first of all in the type. The name is nuclear endosperm. So the formation of primary endosperm nucleus and nuclei are remains free in the cytoplasm of the embryo sac. You know embryo sac. You know all embryo sac consists of cytoplasm. In this nuclei, it is free in the cytoplasm. The number of divisions depends on the size of embryo sac, larger size and more division. These divisions only depends of the embryo sac size. In case the large size of embryo sac is there, this large type of embryo sac, it is divides more divisions. It is normally the divisions also normal. It is mainly the divisions depends upon the size, right? Clear?
Primary
Yes, clear.
nucleus.
So next point, the size of endosperm nuclei are not same. So it is listen carefully. It is what are the statement above statement? The embryo sac, the size of embryo sac and size of endospermic nuclei are the not same. It is too different because the embryo sac is consist
Antipodal.
endospermic nuclei, but these both are the not same. It is the nuclei chalazal end are larger and the micropolar end are smaller. Already we discussed about the embryo sac. What are the embryo sac? What are the parts, major parts of embryo sac? Already we studied in the previous week. Okay, don't confuse. The size of endosperm, where it is located in the endospermic nuclei, in the located on the embryo sac. The endospermic nuclei are not same, and usually the nuclei at the chalazal end are larger, and the micropolar end are smaller. We are discussing in the structure of ovule and types of ovules. We observe these. What are the chalazal end? What are the micropolar? We already discussed. So next, the wall formation occurs subsequently in the glycinemax and arachis hypogea, or nuclei remains free identifiably in the
indefinitely.
Limanthus lugalsi and Acer pseudoplantanus and Maricaria germania. Okay, these are the nuclear endosperms, and hundreds of the free nuclei are found along the periphery of the embryo sac of Primula, Malva, Mangifera, Citrus, Arachis. These are the indicating the wall formation takes place at later stage. There is no initial stage, it is later stage. The wall formation takes place only at 18 to 16 nucleate stage in only some of the species. like Calotropis, Rafflesia, and the Xeranthemum. What are the highest, biggest flower in the world?
Rafflesia, sir.
Rafflesia, sir.
Rafflesia.
Rafflesia, yes, exactly. The wall formation is centripetally. What is the meaning of centripetally, base petally? You know?
Starts from center. Base means
The
outer.
ending of flowers.
Centripetal. What is the base petally? Base petally. The younger one is the base. The older one is the upper.
Outside. Up.
Same acropetally. It is opposite. Acropetally. The old one is the
At top. The smaller one.
no reverse of base petally.
Oh.
So in Coccus nispera, milky or watery liquid endosperms, which fills the large embryo sac. It contains new free nuclei. It is known as liquid syncytium. Besides the free nuclei, the syncytium also contains several multinucleate cells. These cells and free nuclei gradually accumulate along the periphery toward the center and formed in the coconut meat. The coconut meat means it is a white color one. It is edible part. So here endosperm astoria. The cellularization of endosperm is restricted to the micropolar end of the embryo sac. The cellularization of the endosperm is restricted to the micropolar end of the embryo sac. First of all, what is the embryo sac? What are the main? Already I told you. Just I telling about embryo sac. Just you imagine your minds. Oh, these are the embryo sac. These are the parts of the embryo sac. Centrally, it is large nucleus in the antipodals, synergids. So, and also you imagine in the oval structure. What are the chalajal end? What are the micropolar ends? So, what are the degrees? These are the some. So imagine it is very easy to understand this terminology because we are not again not explain the oval and embryo sac. So that's why that day I clearly mentioned it is very important. These are the once you get the all the major parts of the embryo sac and major parts of the oval, it is easily for the further steps. That's why you come there is no these terminology you are not understand. You go to the structure of just you Google search it. Structure of the oval. Structure of the sorry.
Embryos.
Okay. Structure of the oval. Structure once. Otherwise, you come with the flowers also. First of all, structure of the flower. Next, structure of the anther. Next, structure of the
Ovule.
yes. What is this? Next.
Ovule. Ovary.
Ovary. It is oval. Next. Endosperm. Embryo sac.
Yeah.
Okay, types of embryo sacs are there. So these are the link between the all the parts. Just what are the major parts of the oval, major part of the flower, major part of the anther, major part of the what is the stamen, what is the stigma, what is the ovals. Okay, what is the embryo sac? Then we will go. We will easy identify and easy understand these steps. So the new coming to endosperm astoria, the nuclei, the chalajal end are free and embedded in the common cytoplasm. The chalajal phenotypic part of embryo sac grows into the tabular astorium. The tabular astoria found in families like Cucurbitaceae, Fabaceae, and Proteaceae. So I will send lastly in the diagrams. So here in this slide, there is no diagrams. There is no diagrams. I will send you diagrams one by one. So next one is the cellular endosperm. What is this? Next one is the
Cellular.
cellular endosperm. So here
25% families generally
the
die.
cellular endosperm is characterized by the absence of free nuclear stage. Free nuclear stage. The division of the primary endosperm nucleus and few subsequent nuclear divisions are followed by regular wall formation. The first wall is usually transverse, but occasionally the wall laid down after the first nuclear division may be oblique or vertical. These cellular endosperms found in the 25% of the families, generally in the dicards. In the monocards, what is the family members? Only two family members.
RAC and lemonade.
RAC and
Lemonade.
Lemon.
So, in no free nuclear phase at any stage, the wall formation begins with the first division of the primary endosperm nucleus. The cellular endosperms have been further divided based on the orientation of walls. It is the first 2 or 3 divisions. The orientation wall formed after the first nuclear division in the vertical, the longitudinal and to embryo sac. The second wall is also vertical, but the right angles to the first. The subsequent wall formation is restricted to micropolar end. The example is Adoxa and Cetranthus. So the orientation of first wall transfers and one or both cells then divided in the vertically. Example is Scutillaria and Verbascum. The first 2-3 divisions are transfers. Examples is Ericaceae and Aronoaceae. The orientation of the first wall is oblique and the two cells the formed may be equal or unequal size. Example, Myocystis and Arvensis. The orientation of the first wall is identified in the Cincia and Gunera. So here also endosperm Astoria is present. This is the cellular endosperm. It is the endosperm Astoria also under cellular endosperm. The nuclear endosperms also, the endosperm Astoria also there. Here in the different structure of the cellular endosperm Astoria. The occurrence of Astoria is common feature of this type of endosperm. So one or more cells become specialized to function as the Astoria. The Astoria is formed in the micropolar chalajal or at both the ends and penetrate the new cellular tissue to absorb the nutrition.
Nuclear or new cellular.
So I diagram after the is completed these 3 types. I will show the diagrams. So the micropolar Astoria are known to occur in the impatiens of Goili and Hydrocera triflora. The Mangolia abovata, the chalajal Astorium is 2-celled. The first division of primary endosperm's nucleus is followed by the transverse wall resulting into the equal chambers. The divisions in the micropolar chamber are rapid and all the directions. The chalajal chamber divides transversely and a comparatively slow rate. This results in the tail-like chalazal part attached to the massive nuclear tissue at the micropolar end.
Nucleated cellulose. Multinucleated cellulose.
So next one is fallopian endosperms. This type of endosperm is characteristics of the Halobiaceae and confirmed only some monocotyledonous families. The Haemanthis, Gyparanthis, Juncus, Eremerus, Asphodylus. This is an intermediate type of between nuclear and cellular endosperms. The primary endosperm nucleus moves toward the chalazal end of the embryo sac. Here it's divided to form of a large micropolar and small chalazal endosperm chamber. The micropolar chamber, several free nuclear divisions takes place. The cell wall formation, it begins much later stage. The chalazal chamber behaves differently and nucleus may or may not divide. It usually remains phenotypic. These are the regarding halobial endosperm. It is also endosperm astoria. The astoria develops from the micropolar tissue. They are tabular and unicellular, having extensive outgrowths penetrate the nucellate the chalazal end. So here 3 types of endosperms are there. First one is the nuclear endosperms. Second one is the
cellular
endosperm. Third one is the
Halobial endosperm.
So here the mosaic endosperms. It is the one special endosperms, but the endospermic tissues lack uniformity. For example, endosperm of Giames shows 2 distinct color regions, which from the mosaic pattern, the part of the endospermic tissue is yellow and other are the white. This may be due to the failure of the triple fusion. Here, the mosaic endosperms, the formation of mosaic endosperms, it is the failure of the triple fusion. The male gametes does not fuse with the polar nuclei. The male gametes and polar nuclei divides independently, providing the nuclei of 2 distinct characters. It is interspersed during the free nuclear stage. That is the reason why it is in the 2 colors. Just a minute. So this is the reason of in the GMS in the 2 colors, white, yellow and white color. There is no triple fusion. The male gametes does not fuse with the polar nuclei. The male gametes and polar nuclei divides independently. Okay. Hello, dear students. Sorry from some emergency call. So these are the reason. So the maturity when endosperms has become cellular, it gives mosaic and vegetative appearance because of the due to the this reason. Next, one more important endosperm is the ruminate endosperm. The endosperm with uneven or irregularities on the surface is known as the ruminate endosperms. The endosperms with uneven or irregularities on its surface is known as the ruminate endosperms. The ruminate endosperms is found in the Ananoaceae, Maristaceae, Ariliaceae, Ericaceae families. So the ruminate character reflects in the activity of endosperms as well as the seed coat. So why it's come to the ruminate like irregular and uneven surfaces? Because of it is the reflect of the activity. This is the reason. So next, and also the ruminate, it is the seed coat does not form infoldings, but it becomes a consumed and pushed in the irregular manner by the unequal peripheral activity during the late stage of its development and causes seed coat attain and the irregular configuration. So one more is there alluran layer in cereals. I heard about this what in cereals and endosperms is of the free nuclear type. It is generally starchy and dry yet mature by it is fully differentiated endosperms of cereals show transfer cells, embryo surrounding cells, starchy endosperm cells, and alluran layer. One or few outermost layers of the endosperm become highly specialized morphologically and physiologically to form alluran tissue. In barley, this tissue is 3 to 4 layered. The alluran layer cells are alive, whereas the starchy endosperms surrounded by the alluran cells is a dead tissue. The alluran layer protects the endosperms from the desiccation and enables it to survive during the seed drying. The alluran cells are morphologically distinct, different cells. In Ceteria, the alluran cells near the placental vascular supply are columnar and show these cells help in the transferring the substances from the vascular tissue to the embryo and endosperm. So they have been named as the transfer cells. The transfer cells have been Iconocola and Giames, but they are absent rich in rice. They are absent in the rice and wheat. So the alluran cells show thick cellulose walls and non-vacuolated dense cytoplasm. These cells are interconnected by the plasmodesmata. The alluran cells possess alluran grains and spirosomes. An alluran grains is surrounded by a single unit membrane, which is closely associated with the perosomes. The main components of the alluran grains are proteins, phytin, phospholipids, and some of the matrix, the proteinous crystallite body, and non-proteinous spherical body. The globoid are embedded. The globoids present within the globoid cavity contain the phytin and lipids. These alluran layers during the seed germination, the reserve food material, starch and protein present in the endosperm cells, it is digested by the activity of hydrolytic enzymes like amylase and proteases, which are secreted by the alluran cells. The gibberellins have been shown to the activate these enzymes, induce their de novo synthesis. Besides that, the biological importance of the alluran layer is also practical importance. Almost half of the cerebellum is composed in the alluran layers. These are the importance of the alluran layers. The alluran layer, it is only we are seen in the cells. So next, functions of the endosperm. What are the functions of endosperm? An important nutrient medium for the successful development of embryo. The endosperm is the function of endosperm is an important nutrient medium. Nutrient medium means it's provide all the essential nutrients for the development embryo. Right? So that's why the important nutrient medium for the successful development of the embryo. Next, rich in fat, carbohydrates, proteins, which are used the establishment of the seedlings during the seed germination. When it is used during the seedlings during the seed germination. At the time of fertilization, little nutrition available in the embryo sac, but with the formation of angiosperm, enough food becomes available for the developing embryo. Clear? This, do you understand this third point? Next point, at the time of, the division of zygote usually begins after the endosperms is sufficiently grown. Even the zygote and primary endosperm nucleus divides simultaneously. The endosperm grows more rapidly. So the juice of immature endosperms of the plant is used as a nutrition for the developing embryo of the other plant. For example, the coconut milk, it is used as a nutrient medium in vitro embryo culture. When in vitro embryo culture means when the artificially we provide the all the nutrient medium in our laboratory conditions. That time, these the coconut milk is used as a nutrient medium for the in vitro embryo culture. So here, these are the important of and functions of endosperms. Just you just wait. I will share some images of the nuclear endosperms images. So this is the nuclear endosperms. So next one is just you see the slide. These are the cellular endosperm. Next, this is the cellular, nuclear endosperm Astoria, and next one is the cellular. Okay, this one also. So next, these are the cellular endosperm Astoria. So next one is helobial. This one is the helobial endosperm, like this image. This one is the helobial endosperm. So next, ruminate endosperm. This is the ruminate endosperm. Next, alluran layer. You see the diagram. This one, I think, for example, a diagram maize. This is maize grain. So it is the seed gel. It is cotyledon, epicotyl, radicil. These are the alluran layer, like pink color is there now. So this entire part is the starch. This is the seed coat. This is the endosperm. These are the alluran layer. So next we will discuss. This is the development of the endosperms. Next class we will discuss in the development of embryo. Right? Development of embryo. Two difference: endosperms and embryo. What is embryo? Embryo. What is embryo? Yes. Anyone? What is embryo? Yes.
Developing plant from the seed.
Developing. It is more scientifically.
Fertilized zygote is called embryo.
Yes, others? Fertilized? Yes, tell me.
Fertilized zygote.
Okay. Fertilized zygote. So first of all, to develop a plant, what are the stages are there? First of all, yes, any idea? already in the last class, I think, explained. Yes. Yes. You're right. But what about others? What are the zygote? Yes. Are you there? Dear friends, are you there? Yes. No one is there? Yes, please respond. All the students, unmute yourself. Yes.
Fusion of
Your voice is
fusion of male and female gamete is called zygote. The developing zygote, that is the after formation of zygote, it
okay.
develops into embryo to form new offspring.
Okay. Okay. So and one more our topic is embryo development. The fertilized egg is called zygote, and the gametes fusion in the zygote. The zygote is after fertilization, so many divisions, and then finally we got the embryo. Embryo, it is nothing but it is like small plant or small, like what we call in Telugu pindam, right? It is the early stages of the early stage. It is called as embryo. Okay. Otherwise, some before birth. So these are called under embryo. It is very embryo means it is small. It is a fertilized zygote. Fertilized zygote. The fertilized egg is called zygote. The fertilized zygote, it is called embryo. It is the mother cell of the sporophytic generation. The following number of predetermined divisions is zygote is gives rise to an embryo. So zygote, it is gives rise to so many some divisions, reduction, mitosis divisions, meiosis divisions, so many divisions. Then the results in the finally embryo. The development of embryo from the zygote is called anyone? The development of embryo from the zygote. What we call?
Embryo development.
The development of embryo from the zygote. It is called embryogenesis. Embryogenesis. What is the embryogenesis?
Development of embryo from
Development of embryo from the zygote.
Yes. The development of embryo from the zygote is called embryogenesis. So here the dicotyledonous embryo has two cotyledons attached to an embryonic axis. You know, di means two cotyledons. What are the examples of two cotyledons?
Groundnut.
Yes, almost all the cereals, right?
All the cereals.
So what about monocotyledon examples? Mono means single cotyledons. For example,
rice, grains.
Yes,
grains.
Rice and maize,
Mango,
sorghum,
Coconut, papaya.
coconut, mango.
Yes. Seeds, you talk about seeds. Okay. So the development of embryo from the zygote is called embryogenesis. The dicotyledonous embryo has two cotyledons attached to an embryonic axis. The terminal part of the axis above the level of the cotyledons is known as epicotyle, and it eventually forms in the plumule, epicotyle, and eventually forms plumule. The part of the embryonal axis below the level of cotyledons is called hypocotyle. Epicotyle, hypocotyle. What is the meaning of epicotyle? A part of the embryonal axis below level of the cotyledons is called hypocotyle, and it's developed into the radicule. It is embryonic root. You observe the diagram also. The monocotyledon's embryo bears only one cotyledon on a lateral side. So I will show the day after tomorrow. I repeat this also. Just some idea about.
Aileron cells.
So generally, this is the these two are the cotyledons. My slide is visible.
Yes.
So for example, bean seeds. The bean seed under the dicot. So these the right side entire part is the called embryo. These are the cotyledons. How many cotyledons? Two cotyledons. So this above the cotyledons is seed coat. It is the radicule, hypocotyle, epicotyle. What is the meaning of epicotyle?
Cotyledon.
The terminal part of the axis above the level of the cotyledons. Just you observe this one. The terminal part of the axis above the level of the cotyledons is known as the epicotyle, and eventually forms in the plumule, and the part of the embryonal axis below level of the cotyledons is called hypocotyle, right? And it's developed into embryonic root. It is called as radicule. This one, okay? This is the dicotyledon seeds. Once you see the monocotyledon.
Nice.
So here, this one. These are the monocotyledons. Just wait. So here, the monocotyledon, it is consist of the pericarp. It is the cotyledon place. Yellow space is the cotyledon. Coleoptile. This is the endosperms. It is in the plumule. This radical polarizer. Okay, these are the monocotyledon, right? Suppose, for example, you observe the geomes. Same pericarp. It is one of the alluron layer. Is there alluron layer? Pericarp plus seed coat, and these are the yellow part is the endosperm. The cotyledon is the coleoptile, plumella, radicule, colorizer. It is the geomes. So now we will go for embryo. How it is embryo look like?
This
one. This is the dicotyledon embryo. How it is developed? What are the development stages of the dicotyledon embryo? We will discuss in the later. Okay. Just what is the what is the parts, how it is development of dicot embryo. This is the zygote. These are the some stages. Finally we got this type. This is the dicotyledons embryo. Consist plumelle, epicotyle, cotyledons, hypocotyle, embryonal axis, radical is the root cap. These are the, what is this? Dicot embryo, right? Next, monocotyledon. The right side, it is grass of embryo. This one is the monocot embryo. So here just you identify some major parts, just you recognize. We will discuss how the monocot embryo develops, what are the stages occurs in the development of monocot embryo, development of dicot embryo. We will discuss later. So same radical in the root cap. So here what is the difference between the main difference between the dicot embryo and monocot embryo? The epiblast is there, shoot apex is there. So here there is no epiblast, there is no shoot apex. So that just you find out what are the exactly difference between the dicots and monocots. What are the stages? What are the stages of the monocot embryo and dicot embryo? So here this is also very, very important, and also it is 2 slides, monocot embryo and dicot embryo in your practical exams slide. Already permanent slide is there. Yes, please mute. What are you talking? Saraswati, mute. Okay. So these are the some difference between the monocots and dicot embryos. We will discuss later. In this your practical syllabus also, these 2 on your syllabus in the practical, they are given the like slides, permanent slides, already developed slides. Just in the examination, in the slide part, like question A, question B, question C, 1 or 2 slides are given from the embryology. It is very, very important. Either dicot, either monocot embryo. Just keep on a slide on the table. Just you come, and otherwise already adjusted on the microscope. Under the microscope, you go to the microscope. You observe the monocot or dicot embryo. Just identify this type of embryo I identified. Then we will go for the some 10 to 12 points for the practical exams. For the practical, in theory, you in case they are asking in the essay question, all the development stages are you write in the in this part. Okay. Next class we will discuss the more elaborated what are the stages, modes of embryo development in the dicotyledons. So the first stage is zygote after transfer division, the cruciferous type. These also the embryo also many types are there. Aster type, Solon type, Gariophilite type, Chenopodite type. So these are some different types of them. So finally, the embryo development, it is one apical cells, basal cells, the suspensor hypothesis, promaxy distal tier, hypocotyl, basal part of cotyledons, stem cell. These are the some stages of the development of monocot and dicot embryo. So today we learn about the development of endosperms. How many types of endosperms?
Endosperms. 3 types.
What are the 3 types?
Nucleus endosperm.
Cellular.
First one is nuclear,
cellular and haloidal.
Fallopian.
Yes. So here, how much of percentage of in the nuclear endosperms?
Nuclear
56%.
is
What
25.
are the families of?
Angiosperms.
So next one is the cellular.
That is 25% of families of angiosperms.
Okay, next helobial.
19%, that too in monocotyledons.
Monocotyledons. So these are the very important regarding nuclear endosperms and cellular endosperms and helobial endosperms. So today class is regarding this one. So what about the ruminant endosperms? Ruminant endosperms also is very important, and one of the most important. Another one is the
Mosaic
and also alluran layer.
alluran layers.
Okay.
Yeah,
Alluran layer. So what are the main functions of endosperms?
To provide the nutrition. It's a nutrition.
Yes, the endosperms is an important nutrient medium for the successful development of the embryo. It is rich in fats, carbohydrates, and proteins. During the seed germination, these substances are digested and utilized for the growth of the seedlings. Okay. When any plant seeds, during the germination, the endosperms provide that plant
nutrition.
Okay. So next, the young endosperms rich in growth.
Coconut milk.
The young endosperms rich in growth hormones also. Growth hormones. What are the growth hormones?
Gibberellins and auxilins.
Gibberellins, axins, and cytokinins. What are the role of these hormones? What is the hormones? What is the plant hormones? Any idea?
Which activate or works as a catalyst to grow or speed up the development.
Okay. So this is the functions of endosperms. It is the plant hormones. It is growth and development purpose. It is naturally produced by the plants, some of the fungi also. So some of the fungus produces the gibberellins. What is the fungus name? Any idea? Gibberella fusicorae.
Mycorrhiza, sir.
Gibberella fusicorae.
Okay.
Mycorrhiza not production of the plant hormones. So the young endosperm of mice contain the cytokinin called zeatin. In the coconut milk and extract of corn are frequently used as a nutrient in the embryo culture. Already I told you. The milk, coconut milk. The coconut milk also, it is used as a, so once so here, once you observe, it is clear?
Sir, please send pictures in the group.
Not clear. Okay.
Sir, send pictures in the group.
Sure. After completion of all the syllabus, just anyone, please send me one message. Then I will send you. So not now. So this is the mature coconut fruit. Mesocarp, it is like husk in the upper part, right? So in the epidermis. And endocarp. Endocarp, it is the shell. After the like this is second part, right? Endocarp. What we called is endocarp. So cavity with coconut water. These are the coconut water, and these are the endosperm, like coconut meat, white color wala. Okay, white is there na. It is endosperm. How it is appears? It is the water, the coconut water. It is nothing but the endosperm water, right? These
Liquid.
water used as a
Nutrition.
nutrient medium in the embryo culture technique in the in vitro conditions, right?
Condition.
Clear? Do you understand clearly?
Yes. In vitro.
Yes.
Yes, sir.
The coconut milk or coconut water used as a nutrient medium. It is used in the nutrient medium, the embryo culture. The embryo developed artificial in the laboratory conditions. That time we are using the endosperm as a coconut milk or coconut water. So the endosperm regulate the precise mode of embryo development. In the absence of endosperms, very young embryos often fail to mature even in the richest artificial nutrient medium. So this is the some functions of the nutrient. The functions of the endosperm. Okay. So next class we will discuss the development of the embryo. So how many topics are remains? So once completion of the development of embryo, next apomixis. What is the meaning of apomixis? Any idea? Yes, apomixis. Yes, anyone? Yes, apomixis. Yes. Next topic is the polyembryony, and the block fifth is the embryology of angiosperms in the third, parthenocarpy, experimental embryology, application of embryology in taxonomy, agriculture and horticulture. Application of embryology in taxonomy, agriculture and horticulture. Next one is, last one is principles and application of palynology. What is the palynology? Any idea? Yes.
Study of pollen grains.
Study of
Pollen grains is called palynology.
Pollen grain.
only pollen grains or other any other? Yes.
Structures and the
Seeds విత్తనం.
and their uses, sir. Some pollens are used as medicine also, no? So
No.
the uses of the pollen grains also comes under palynology.
Simply, it is the some morphological features of pollen and spores is known as the palynology. So this is the scientist Williams and Hyde is deduced from the Latin word pollen, which means the spread and distribute. What is the role of generally pollen grains? It is the distributed. So it is the pollen grains. So it is transferred to the one plant to another plant. Okay, to the fertilization purpose. So before that, what is the parthenocarpy? You know parthenocarpy?
Parthenocarpy is the fruit developed without proper fertilization. False fruit, I think so it is.
Okay. What about others?
Seedless fruits are called parthenocarpic fruits.
Okay. So in we will discuss in the palynology also. It is also very important in your future. In future, in case any research in the palynology also, one specialization, only specialization is available in the palynology specialization in the University College of Science, Usman University. It is the college is located in the Saifabad. The specialization is available in the botany department. So that is Paleontology department. Okay. So study of fossils.
Fossils are there.
Fossils.
Paleobotany.
Paleobotany. So, and any idea how many types of paleontology? Aeropaleontology. Any idea? Melitopaleontology. Any idea? Melito. Melitopaleontology. Any idea? Nectar. Nectars. How to produce honey? Honey. How to produce honey? Honey production. Yes. Yes. How to produce a honey? Honey bees collected
Nectar from the flowers, and they will convert it into honey.
Honey. So that is also melitopaleontology. Okay. Melitopaleontology. So here, these are some important topics. So our last class, how many class remaining? How many class?
7.
I think the last class is 27th or 26th or 28th. 27th. My paper.
6. 6 classes are there, sir. 27th is the last day.
Okay.
Embryology. 15th, 17th, 20th, 22nd, 24th, and 27th.
Okay. So when the honeybees collected in the through the pollen grains, so when we will identify the honey, some different processes are there. These type of pollen grains found on the honey. So the research, which pollen grain it is consist of which plant. This, suppose for example, this pollen grain, this pollen grain, it is belong to this family. Suppose the round pollen grain is there, na? This one is acacia. Acacia. So these are the pollen structure also there. So our Kumar sir is there, na? He is working on these pollen grain studies from the honeybees.
Okay.
So we are collected in the different honey samples. So, and after the some procedures are there. Then finally we got the pollen grains. So that means we find out suppose this particular area, these flowering plants are there. These flowering plants, the honeybees collected honey from these different plants. So this area, these different plants available in this area based on these pollen grain studies. Okay.
Okay.
So these are the pollen grains.
Different shapes.
Yes. Do you understand? This is the under melitopaleontology. This is under the These are the pollen grain structure. We will discuss in last how pollen grain structure, how it is surface are there, how it is arrangements of eggs and intent structure. We will discuss in the later. But these are the good research going on in our Usman University. So this is also. So how is it is beautiful in the pollen grains? Very simple technique. We are identify suppose these pollen grains are there. These pollen grain found in these plants. So the honeybees collected from the different plants, they are collecting in the one place. Then we will collect the honey. Then we will go for analysis. Then we will find out these pollen grains. Based on these pollen grains, what are the plant species represent in this area? We find out easily in these studies. That is the melitopaleontology, right? Thank you. Thank you, all of you. Hello.
Thank you.
Students, thank you.
Thank you.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you.